covered some of the basic ideas in sampling design, I want to briefly talk about some considerations when using plot samples for sampling items. This is only one way that you could do it. So obviously, depending on the variable that you're measuring, you might want to use something else. But it is a technique that's used commonly, and some of the ideas we're talking about would apply to other approaches as well. Just briefly talk about some different ways of collecting samples. Oftentimes we go out and collect data from plots or quadrats. These are usually small geographic areas, anywhere from less than a square meter up to maybe a hectare or two. Commonly used to sample habitat or count animal numbers. Another approach is to go out to points and take measurements from a set of points established throughout the population. We often do this with measuring tree density or tree basal area. Or transects are another approach that are often used, where you take a straight line across your region of interest, and as you walk that transect, you may measure something like vegetation, what kind of plants intercept that transect. If you're interested in animal abundance, you may count animals adjacent to the line. In that case, you probably want to use something like distance sampling to take into account animals that were missed. But a variety of different approaches that can be used for sampling populations. Now let's just focus in on plot sampling, since that's what's often used when conducting the kinds of studies we're interested. 4 questions that you really need to think about whenever collecting data from plots. How do you determine the location of the plots? What shape should you use? What size should you use? And how many plots should you use? Well, for plot location, use one of the sampling designs that we discussed earlier: random, stratified, clustered, etc. So I won't go into more detail here. Plot shape. One of the things you want to do is minimize edge decisions. If you're counting vegetation and you're trying to determine whether a particular plant is in or out, that can often lead to error. So if you minimize edge, those kinds of errors will tend to be minimized. And we know that circles have less edge per area than squares, which have less than rectangles. On the other hand, when organisms are aggregated, rectangles often give better variance estimates than square plots. Because if you think about it, if a square or circular plot lands right on top of a clump, then you're going to get a high count in that plot. If you happen to land in an area where that species is not present, you're going to get a 0. And so you tend to have a lot of variation from one plot to the next. Rectangles tend to reduce that because they're more likely to include some portion of a clump of individuals, and therefore the variance overall will be reduced. Plot size, you want to minimize edge decisions. So large plots are better than small plots. You have fewer individuals along the edge relative to the total number of individuals you're sampling. If you're out counting trees and you have a big plot, 95% of them are well within the boundaries. You really don't have to make any call. And then there might be a few that are right on the edge, and you determine is that the center of that tree, inside or out of your plot. It might be a little bit more difficult. Larger plots, that kind of variance is going to decrease. On the other hand, you have counting bias in large plots. With trees and animals or things that don't move, generally that's not much of a problem. But with mobile organisms in large plots, you tend to underestimate the total. When you have aggregated organisms, you want to choose a plot size that's either larger or smaller than the average cluster or aggregate size. Rules of thumb, plots should be large relative to the size of the organism being studied, and you should target your plot size to average at least 4 to 10 organisms per plot, and that way your variance will tend to be reduced and you'll get a better estimate, more precise estimate. And as far as number of plots, again, using the sample size approaches that we talked about, it really depends on the level of precision and the power of your statistical test and those factors that we've already gone over.